Hello, welcome to A Moment of Science, please. I'm one of your hosts, Ana.
And I'm your second host, Margarita.
Today we have Dr. Mortha on our podcast, who is a professor in the Faculty of Arts and Science within the Department of Immunology. Welcome, Dr. Mortha.
Good evening, you two. Thank you for having me.
We are delighted to welcome you too. We would like to begin with your background. If you could please introduce yourself and tell our audience a little bit more about you, your background and education, and what really interests you about the discipline of immunology.
So my background is I'm a trained biologist that decided to study the immune system. So I was born in Germany. That's where I received my education. I went to university in a small town called Erlangen, which is it's a tiny little university, and it has. I initially started to join at university to become a high school teacher. So I started to study biology, zoology, botany, all of these basic disciplines, and chemistry. And then I took a couple of lab courses and decided and read a couple of textbooks and found out that there is so much more to learn than just basic zoology and botany. Trying to understand cells, trying to understand the machines they use to fulfill their functions. And that got me really excited into more molecular biology and genetics. And also biochemistry. And so I then decided to shift gears a little bit and focus my studies more on those molecular topics. That was, it was a little bit tricky. I had to study and finish both degrees because in Germany there is an option to get sort of a student loan when your parents are not able to support you fully. So I received one of those loans, and but you pick basically one subject or one degree that you would like to get, and then you have to finish this one. Otherwise, you will lose that support. So I could not afford that, so I had to study two things at the same time. And then I did basically the degree for the high school teacher with all of the chemistry courses and basic biology courses on the side. I did all of the molecular courses, microbiology, biochemistry, and then there was an option to hear immunology lectures. And I was sitting in the first immunology lecture, and I was completely overwhelmed by the complexity of that subject. But I think it was the teacher that gave the lectures. He was very engaging and very enthusiastic, and that kept me motivated to learn more about that topic. And then there were journal club courses that you could take, where you had, the professor would hand out a landmark paper, and then you would discuss this with a partner in a team. You would present that paper to a larger group, and the larger group was able to implement what they learned about immunology into this particular paper. And that professor did a really, really good job. It was a small group course, which allowed you to speculate, and it allowed you to really discuss and even come up with wild theories. And the professor who gave that course, I mean, he's still doing cool science, but yeah, remember that those were really, really nice courses, which kept me motivated to continue. And then I decided, okay, I need to do more lab courses. I need to get into labs that do immunology, or at least biochemistry or something molecular. And then I took all of the courses that I could get into to learn the necessary methods, your basic biochemistry methods, protein purification, cloning, and then I was an opportunity to get into a course that was over several months, and that was the opportunity that brought me into immunology. So this was in an immunology lab that was studying the development of B lymphocytes, which are the immune cells that produce antibodies. And the development of B lymphocytes is that they have to generate these antibodies, and not every antibody is distinct. And so in that process, there has to be, because the antibodies can react against your own body and against your own proteins, so there has to be selection processes and basically quality control implemented in that developmental process. And those steps are important to avoid autoimmunity and autoreactivity of these B lymphocytes. And so that lab was studying one of those processes, the first process that controls the assembly of the building blocks of these antibodies. And so there was a nice combination of protein biochemistry and some cellular immunology. And from then on, I was able to do back then it was called a diploma thesis, but it's the same as a it would be a master equivalent, something like that. And so then I finished with a master's in immunology from that laboratory, working on the developmental processes and the checkpoints in the development of B lymphocytes. From then on, I of course had to decide whether I do a PhD. And instead of staying in the same lab, I decided I need to learn something more. There's so many more cool immune cells. And one thing that motivated me, I saw I got a job in a laboratory after I got my diploma to work as a sort of a research technician. So I had 3 independent projects that I was taking care of, and I only got into that lab because I worked as an undergrad student for that PI to organize his files and fed patient information into a database. So really, a boring job that I was doing somewhere in a basement, but it opened the door. Yeah. And so with that PI, I started to look into T lymphocytes, which are another important part in our immune system. So these lymphocytes have, again, a receptor on the surface that is very specific. It recognizes foreign proteins. And those immune cells can also recognize virus-infected cells, and they are very good in instructing how our immune system will, well, basically, they dictate the strategy our immune system will take to fight a pathogen. And this professor was very interested in studying HIV and CD8 T cells, which are T lymphocytes that can actively kill and destroy virally infected cells. And I thought, that's super cool. To study a cell that can kill another cell, I want to do that. So I got this job and started to work in his lab, completely independent. I think the only person that was in his lab as well there were two lab technicians, and they showed up and did the diagnostics for that lab. But yeah, everything else, it was basically an open playground for me. And meanwhile, but this was still close to the same university where I did my training. But meanwhile, I started to look for opportunities to do a PhD. And back then, there were no graduate programs like nowadays. Sounds bad. But you would basically write to a professor at the university and ask, could I become a PhD student in your lab? And you would not go into a program and get all of the additional soft skill trainings and courses available. And so that's a little bit different than to the US system right now. Nowadays, they have graduate programs in Europe all over the place because it's a good system. So I decided to reach out to professors, and you both probably know how hard it is to get a response from them. So some of them never replied when I sent my emails, and I was trying to personalize them as good as possible. I was reading all of their papers, and I was trying to figure out what really excites me about what they're doing. And I was trying to even propose tiny little projects, say, I would be very interested in understanding that and that aspect of this and this cell type. And what was really understudied back then was the innate immune system. So people knew that an immune response requires two arms, an innate and an adaptive immune response. And the innate immune response is the response of the immune cells that happens very fast, very early after an infection. And you guys probably know, if you catch an infection, you'll start feeling very sick for about a week. And that's when it starts to get better. And by that time, your adaptive immune system is activated and can start to eradicate and eliminate the pathogen. But that again takes a couple of time. And without the innate immune system, the activation of the adaptive immune system would be delayed even further. Excuse me. So I decided to do a PhD on the innate immune system because I thought that would be something super understudied and I need to understand this better. There is a lot of things to explore. And the only innate immune cells people knew back then were natural killer cells. So those were cells very similar to CD8 T lymphocytes. They were capable of killing cells. And back then, people just started to show that these cells can kill tumor cells, blood cancer, for instance, or predominantly. And now those cells are used and genetically modified to selectively target specific blood cells and eliminate them. So yeah, they've come quite far in terms of using those cells then therapeutically. But back then, that was what was known about those cells. And I got an email from a professor back that I contacted who was working on that particular aspect of natural killer cells. And he invited me for an interview. I gave a presentation about my diploma work on B lymphocyte development. And he showed me some preliminary data on natural killer cells in the intestinal tract. And back then, the only organs that I studied were the thymus, lymph nodes, and the spleen. So the major, immune organs that everybody knows. But to really look into the gut, which is a massive organ, even inside of a mouse, to look into these organs and isolate immune cells out of that organ and then to characterize them, that was something that already excited me a lot. And he showed me a set of data where he showed a population of NK cells, and he used a transgenic mouse that was allowing cells to report the expression of a transcription factor. And within the population of NK cells, he found another population that expressed that particular transcription factor, basically indicating that NK cells are not all the same. There is another population of NK cells that have a specific transcriptional program active that might make them different. And I thought, okay, I need to understand what those cells are, because that was basically a new lymphocyte cell type. And if you identify something like that, you should jump on it and should start to understand those cells. And that was what I decided to do then. And that's how I ended up studying innate lymphoid cells, because it later turned out that that particular transcription factor is important for the differentiation of a group of these innate lymphoid cells. And so, wait, just serendipity, yeah? Just right time, right place, right data slide. Yeah,
That's really
after
interesting.
Sorry.
It's really interesting how one teacher sparked you to do such a massive research and continue to explore the science and to explore more. It's really interesting.
One prof sparked a whole series of events which led to where you are today. And I think your journey really highlights that it's normal to change career paths. As you said, you were starting off as a high school teacher, but you decided to go into into science rather than into teaching. And I think this is something most people have trouble really comprehending or acknowledging, since most people, once they decide on a path, they kind of want to stick to it. So it's
The interesting thing though is that you should not not necessarily you should continue to evolve and look into new elements. When I decided to do a postdoc afterwards, I decided to reach out to laboratories that would study myeloid cells, mononuclear phagocytes, because I found that these innate lymphoid cells can produce a factor that can communicate, allow those cells to communicate with myeloid cells. So instead of continuing working on these innate lymphoid cells predominantly, I decided, okay, I need to see what they can do to other immune cells and how they can maybe modulate immune responses. And instead of going to a lab that would study innate lymphoid cells, I decided to go to a specialist that studies mononuclear phagocytes, myeloid cells, and started to develop basically a new type of program. And this is something that we we continue doing here afterwards as well. So we continue to study the interactions of these ILCs and mononuclear phagocytes here in Toronto.
yeah, wonderful. Thank you for sharing. So actually, we're going to move on to our next question, which actually mentions, starts talking about the paper that we want to discuss in this podcast, which does have some mentions of mononuclear phagocytes. So one of your most recent papers, in which you are a co-author, is titled Microbiota-Dependent Crosstalk Between Macrophages and ILC3 Promotes Intestinal Homeostasis. This paper focuses on tissue-resident mononuclear phagocytes, or MNP, which consist of two different cell classes, specifically the macrophages and dendritic cells. Since not everyone in our audience is familiar with immunology, can you please explain how the MNPs work and the differences between the two cell classes considered MNPs?
So I hope I don't get into trouble for that. But some people will, mononuclear phagocytes is a large term that summarizes cells that are phagocytic, so they can eat and engulf particles and proteins. And one of the things that is important for the immune system to do is to surveil and get an image of the antigens, the foreign particles inside and on your body, and basically evaluate, are those things that I can tolerate, or those things that I should attack. And to do this, you need to have cells that survey the tissues and the surfaces and are capable of sampling these antigens. And then they need to ask, because those are not the decision makers, they're basically sampling these antigens, and they're asking for feedback from T lymphocytes most of the time. And those T lymphocytes will tell them, okay, this is something that I recognize, or this is something that I've seen before. And if the T lymphocyte has seen it before and has decided this is something that we can tolerate, then usually the body doesn't react against those antigens. However, if that antigen is presented to a T lymphocyte and that T lymphocyte will start to be activated, then that can result in an immune response and an inflammation. Of course, this is very simplified procedure. Yeah, it's a little bit more complex than that, but basically these mononuclear phagocytes are the ones that can sample and sense the environment on mucosal surfaces or in other organs, and they basically decide if immune response turns out to be tolerogenic, something that we just accept as being there, or something that we decide to not tolerate. And it really depends on how these cells are programmed. And so the key difference between these mononuclear phagocytes that you refer to, dendritic cells and macrophages, is their origin. Both come from precursors, blood precursors, and hematopoietic stem cells are the shared precursor, and then they branch off into a lineage that will give rise to dendritic cells, and a lineage that will give rise to macrophages. Macrophages are migratory cells, and they eat a lot. But they are not very good in leaving the tissues and then going to the places where the lymphocytes are. Dendritic cells can do that job much better. So what both can do is they can eat in phagocytose, and one of these cells, the dendritic cells, will take that cargo and migrate to a lymph node where they can talk to T lymphocytes and tell them, okay, is this something that we should react against or not? Macrophages, on the other hand, are eating as well, but their property is not too much taking these antigens and bringing them to a lymph node. Is more cleaning up, more the janitors of a tissue. So a dying cell, for instance, can be engulfed and cleared by a macrophage. There was some diseases in the brain, in the brain, for instance, where this process is quite important. Where the macrophages in the brain, the microglia, are important to maintain neuronal homeostasis. And so these two have, even though both of them have the capacity to present antigen, one will do that in a tissue, and one will execute a function in the tissue, the other one will leave the tissue to instruct the immune response. Now, both of these cells in that particular paper share a receptor for a growth factor called CSF2 in this paper. It stands for colony stimulating factor 2. And what this factor does, it promotes the survival of those cells, and it promotes an effector program. And in a healthy intestine, this growth factor can program these cells to tell the T lymphocytes in the lymph nodes to tolerate an antigen. So they will come with an antigen to the T lymphocytes, and if they received GM-CSF or CSF2 signal, they will additionally tell that T lymphocytes, look at this, this is not that bad. We should not attack this. And this will result in a tolerogenic response. And so if that particular growth factor is absent, and the cells that will induce a tolerogenic immune response are obviously impaired in their development. And what we found was that these ILC3s, these innate lymphoid cells, are a predominant source of this CSF2. So they would instruct the dendritic cells that are inside of the intestinal tract to adapt a phenotype that tells them, okay, please don't let us attack this antigen through the production of this growth factor. What they also do on the macrophages, because that factor acts on both, is they mediate the antimicrobial host defense. So if a macrophage receives stimulation through CSF2, they are more capable of phagocytosing and eating and destroying invading bacteria, for instance. So they're better at defending the intestinal tract. So it plays an important role in modulating these mononuclear phagocytes.
Wonderful. Thank you for clearing that up. I think for everyone in the audience that is looking into reading this paper, I think this will definitely help clear up the paper for those that do want to read it, because there is a lot of background knowledge that is usually required to comfortably read a paper in science. Therefore, thank you so much for clearing that up. I think it is very clear, even for someone that doesn't know much about immunology. I think they'll have a much easier time reading this paper now.
Exactly.
Thank you again for bringing out more knowledge about this paper, because our next question is also related to this paper. One of your finding was that CSV2 deficient mice had decreased amount of regulatory T cells in the colon, but not in the small intestine. Can you please explain why the regulatory T cell amount decreased only in the colon, but not in the small intestine?
So a regulatory T cell is a T cell that tolerates an antigen and will dampen the activation of T lymphocytes that would like to react against a specific antigen. So they're very important to tame the immune system. Now, the reason why these regulatory T cells are regionally distinct or differently affected is that so far people have not started to look into the regional differences of immune cells across the entire intestinal tract. We know there are specific compartments within the intestine where lymphocytes or leukocytes can reside and can communicate. And we know that there is specific anatomic features distributed across the entire intestinal tract. This also coincides with the functions of the intestine at the different locations. The small intestine would mostly be there for nutrient absorption, whereas the large intestine would be more for water absorption. And so this already at the anatomical structure of those organs, you can see that these are specialized regions within or across that organ. And so would probably be the function of the immune cells in specific locations. And there was one publication by a group from Weill Cornell. They demonstrated that in the small intestine, these ILCs can produce another cytokine that is important in supporting these regulatory T cells. So there could be compensatory factors or regional specific effector functions by those cells that can then, through specific mechanisms, locally generate the same effect, right? I think this would biologically also make sense. You would not want to use the same system to generate the same cell type. You would like to have backup systems, maybe.
Thank you for elaborating upon the mechanism. I know when I read this paper, this kind of confused me. I'm like, oh, why exactly? I think this is a really reasonable potential mechanism. So thank you so much for explaining that part. I'm sure, again, the readers who will be reading the paper after this or with our podcast will probably have a much easier time understanding the potential this specific finding. So your study described in microbiota-dependent crosstalk between macrophages and ILC promotes intestinal homeostasis required the use of mice. What are the advantages and disadvantages of using a mouse model compared to other available models, such as cellular models in this scenario?
That's a good question. So ideally, this is, of course, the use of mice is hard to justify. These are, there is economical reasons for that. There is reasons that allow you to extrapolate results into humans and can say maybe it's the same. It is, of course, a cellular system is really nice if you can get this working, but most of the time this is really difficult, especially for complex organs like the intestine. And then you always want to consider, is the cell type that you use in a cell culture dish actually recapitulating what you're seeing in vivo in a live animal or in a live organism? And so the mice are, scientists have learned to modify the genome of mice relatively fast. There is a lot of genetically modified animals available that allow researchers to study specific effects of genes on the organ or the cell type or the process of interest. And so we in this study made use of specific knockouts. Those are the mice that are lacking specific genes, where the gene has been destroyed, to understand how factors within the immune system interplay. So the really nice thing about animals, for instance, is that you can have them in a genetically modified state where a gene is absent, and you can try to bring the cells or the product of that gene back into that mouse, and you can see, does it restore and does it change the behavior in the disease? Does it make the disease better, for instance? So it is also a good way to test the potential of a protein as a drug. And so we use a lot of mice in specifically immunology research, not only because of the interplay of cells, because I already mentioned mononuclear phagocytes, which again are two different cell types, and then I mentioned innate lymphoid cells, a factor that they can produce, and I mentioned that there are T lymphocytes. So those are a lot of elements that will be hard to control in a cell culture dish. And so in that case, using animals is a better way to understand complex systems like that. If you're very interested in understanding a cell intrinsic process, that's a different thing. In that case, you can definitely use a system where you have a cell line, for instance. So for ILC3s that we study in the lab, we're using a cell line that recapitulates those cells quite okay. Those have been published by a researcher here at U of T. His name is Jim Carlisle. He has generated those cell lines, and those are immortalized almost completely ILC3s. We can use them to knock out specific genes in a cell, and then we can study the biochemistry and the function of that cell in isolation. And if we observe a nice phenotype, we can still try to use and see if someone has already generated a knockout animal, or see if there is a patient population that has a genetic defect, for instance, in that gene that we targeted. So those are the reasons.
Wonderful. Thank you for explaining those. Obviously, there is some controversy around using animal models. However, I think your explanation of the importance of in vivo system, I think it definitely justifies it at the moment, since we cannot do whole scale systems just from cells, unfortunately, at this point.
So the next question is going to be about Crohn's disease. Crohn's disease is a type of inflammatory bowel disease. It causes inflammation of your digestive tract, which can lead to abdominal pain, severe diarrhea, fatigue, weight loss, and malnutrition. We were interested in how the results of this study are significant for further research about Crohn's disease.
It's also a good question. So overall, Crohn's disease it's quite severe disease. There is currently no cure for that disease, and the treatments, they're long-lasting. They're very expensive, and they're not always working in every patient. So there is patients that in some therapies just won't work. And the other thing that makes this disease particularly interesting is there is heterogeneity in reasons that can drive that disease. And a lot of the research that has been done in the past has looked into genetic defects that can trigger the onset of this disease. Now, not all of the genes that have been identified to be associated with that disease will cause this disease spontaneously. So there is additional factors like the environment or the collection of microbes that live inside of our intestinal tract, the microbiome. Lifestyle, for instance, dietary changes, have been all implicated in driving this disease. So it is quite complex, and if you find a gene that's associated with that disease, then it's probably not going to affect all of the patient population. Now, in Crohn's disease, for instance, groups of patients have been identified that carry mutations in the receptors for CSF2. So those patients, for instance, the mononuclear phagocytes cannot respond properly to that factor that's produced by these ILCs. And they will not adapt the right phenotype. For instance, macrophages will not be able to phagocyte host bacteria and eat them and kill them. So their ability to defend a person with that genetic defect against a microbial infection is significantly impaired. The other thing that has not been shown yet that could be affected is the ability of patients with this mutation to generate regulatory T cells, for instance. Another thing that has been identified, and it's not genetic mutations, is the presence of antibodies in a subgroup of Crohn's disease patients that binds to GM-CSF and can neutralize the function of GM-CSF. And that is obviously something that doesn't necessarily have to be genetically determined. So for some reasons, some of those patients start to develop antibodies against this growth factor that they need to reprogram their mononuclear phagocytes in the intestinal tract. And what the result is very similar to the genetic defect that I just described in those patients. So their mononuclear phagocytes will not be able to kill and defend the host, and they will not be able to adapt that program that they need to generate an immune tolerance. So in these patients, they clearly have a phenotype that is almost 100% identical to what you found in the animal model in mice. So here, this is a nice example where the studies in the animal actually recapitulate the humans, and where the mouse actually turns out to be a good model to investigate how to intervene and how to maybe come up with therapies for that group of patients.
That was interesting to know that the mouse model is so applicable to the human for Crohn's disease, and it has really good potential to have a better treatment in the future.
Yeah, and the fact that Crohn's disease can have a lot of different factors contributing to it in different patients is also very interesting. It's definitely one of those more complex diseases, which is not just like one gene, but a lot of different factors contributing to it. So as you know, every study does have limitations, and we were wondering if you'd be able to identify any potential limitations in the study we discussed today.
Well, one limitation is that you cannot 100% recapitulate the entire study in humans side by side. So that is one thing. The other thing that makes it very hard is to be cell type specific. So what you would really want to show is that the CSF2 that comes from a specific cell type is sufficient to control the entire process, or not. Right? And that is one of the limitations to address the question if the ILCs are sufficient to drive the to drive that process. We had to use mice that are, we had to basically reconstruct in a mouse parts of their immune system to see if these elements interplay to induce immune tolerance of regulatory T cells. And that, I think, is, it shows the process applies and works, but that is, it's very artificial to do that. And there is different ways of addressing that are maybe a bit, they would involve, generating more transgenic animals that are more specific, where the entire immune system develops in the same time, the same fashion, instead of using a mouse that is bit by bit rebuilt in terms of the immune system to study the interactions of those cells. That is something that I would have really loved to include in that paper.
Well, hopefully you'll be able to publish it in the next paper, and it's definitely great that you were able to identify limitations. It is sometimes a bit difficult for people to do, but hopefully these limitations are overcomable later on as science does continue evolving.
I think what is really interesting is to look into the unanswered questions. What are the things that you would want to address next in that particular mechanism? Because the thing that I haven't mentioned yet is the integration of the microbiome in driving this process. And that, I think, is something that is not yet well understood enough. And so this is an active area of research in the laboratory. We're trying to figure out what are the signals that the immune system, specifically those mononuclear phagocytes, have to receive to kickstart the generation of the tolerance.
Limitation bring out the room for progress in science. And today we heard about your background and how you end up in immunology. And we would like to ask as a follow-up question is, do you have any tips or advice for students that are interested in pursuing research?
Yeah, immunology is the best subject. No. So, of course. Well, in theory, every cell is an immune cell, if you think about it. I find it very interesting because the immune cells are involved in almost every process. If it comes to development, if it comes to maintenance of tissue cells, you'll find an immune cell involved in almost everything. So that's why I think this is a really nice topic. More generally speaking, for students, one of the experiences that I've made is that persistence pays off. You have to be almost annoyingly persistent. I remember that there was this biochemistry prof, and I wanted to work in his lab. And I don't remember exactly which year I was in, but still not experienced enough. But I was trying to get a job in his lab. And I think every month, at least once, I stopped by his door. Back then, you would not send too much emails. And I would ask, hey, it's me again. Do you have a lab job? Maybe not, in a more formal way, of course. Yeah. But no, at some point he would say, you know what? Come join this journal club course that I give, and it was very similar to the journal club that I explained, and we were basically discussing papers, and so that was my chance to leave an impression. There was, not just once, but I could do this for an entire semester, once a week. And so, of course, I was always prepared, and in the end, yeah, he offered me becoming a lab assistant in his lab, and I was then able to basically tutor courses for undergraduate students and do some of the experiments. I think this was, but the bottom line is sometimes it doesn't work like this all of the time. Yeah, and in the end, as you can see, I didn't end up doing biochemistry. So not, the path might not be straight all of the time. Yeah. And I would use the opportunities.
Yeah, I think most people that have gone through the research process would agree. From my experience, I also agree that being persistent does really help. I sent up like 3 follow-up emails in addition to a first email before I got a position. And you're right, it is because persistence, and I think this is coming from you, will encourage more of our viewers to hopefully be persistent. Because if a prof confirms that like annoying persistence is okay and is, well, sooner or later appreciated, I think it will definitely motivate people to continue with that.
I just want to clarify, maybe it was not the persistence that convinced him, but more the engagement afterwards in that course, right? So I think it's not only sending out 10 emails to the same email address. I think it is also basically conferring the interest and excitement, and the motivation and the drive. And this is something that I think is important as well. You have to somehow show that that's the thing you want to do. And so I would try to include that as well.
For sure. So hopefully for all our viewers, if you do find something that you're really interested in, you have to show that to the prof you're emailing and or calling or stopping by at office hours, whichever method is going to be applicable once you guys are watching this. So now we're getting towards the end of our podcast. We usually finish it off with the question, who is your favorite scientist and why?
Yeah, that question I like as well. And I try to think about one, but I can't think of one. I think there's more groups of scientists that I like a lot. So first of all, those that have influenced me most are all of those PIs that I've worked with in the past. Those that trained me, those that were mentors, and you experience the bad things about it, you experience the good things about it. Yeah. And then you can try to take what it needs to become, what you think is important to become a good mentor yourself and a good scientist as well. And a lot of that is motivation and is persistence. And even though experiments might not work out, that's one of the things that I learned from them, is to stay motivated, stay positive. So those two are some very good examples of teaching me how to stay motivated and excited about science. But then there's others that I admire a lot because of their boldness and their. There's one example. Her name is Polly Matzinger, and she is maybe I don't know, one of the most important immunologists, I think. She came up with a model called the danger model in maybe mid-90s or something. And that was completely paradigm shifting. And she would continue and persist on that theory. And this theory has now been proven a bazillion of times. But back in the mid-90s, she received not so much love for that theory. So that type of sticking with what the results tell you is really good. That's one person that I think you can look up to. But then there's others that propel science forward because of the technology and because of their the ways they transform science. Some of them are some scientists, for instance, there is Stephen Young is a very good scientist, and Boris Reiss. Those two have generated very important transgenic mice that are now used all over the place to study mononuclear phagocytes. Without those two mice, without of their efforts to generate those mice, a lot of what we know now would not be possible. So I think those type of scientists that if they have a question and they want to answer it, they just build the tools to address it. Those are very, very good. Another good example is Ido Amit and Aviv Regev. Those are two scientists that spearheaded single-cell RNA sequencing. And back in the early 2000s, they had, 2010, starting onwards, they had a couple of very transformative papers looking at the transcriptional changes within individual immune cells across organs or across populations of dendritic cells or mononuclear phagocytes. And so I think this technology, very similar to CRISPR, has been changing how the field has evolved. And I think engagement in driving technology forwards to the to even further limits, something that I think is very, very good. And then there are those scientists that sell really good stories. They're very good in writing papers. And I think the best example is a person called Yasmin Belkaid. The papers that she writes and the presentations that she gives they're amazing. And the biology that she studies it's always new. It's always something that you would have never expected. And I think those are the scientists that motivate me.
Thank you for, I would say, a more unconventional answer. I think you really hit all the bases for the different scientists' strength and a variety of topics. So thank you so much for sharing. And so with that, Dr. Mortha, thank you so much for coming on today and sharing your research with our audience. It was a pleasure to talk with you. For the audience, this concludes our podcast. Thank you to everyone who has joined us. If you found this podcast engaging, keep an eye out for our next episodes, and don't forget to subscribe to us on YouTube and follow us on our social media.